The African Hito, also known as the African catfish or the Clarias gariepinus, are extensively cultured in the Philippines. It is popularly in aquaculture is due to its relatively fast growth rate, adaptability to various environmental conditions, and tolerance to poor water quality. The aquaculture of African Hito provides livelihood opportunities for many people in rural areas, and it contributes significantly to the country's fish production. Introducing African Hito to the Philippine waters has raised concern about its potential impact on native aquatic species and ecosystem. While it offers economic benefits, there are also environmental considerations such as competition with native fish species and potential disruption of local ecosystem. In fish anatomy, terms dorsal, ventral, anterior, and posterior are used to describe different orientation or position on the body. Dorsal, these refer to the upper side or back of the fish, the dorsal fin, for example. Ventral, this term describes the lower side or the belly of the fish. The pelvic and the anal fin are typically located on the ventral side. Anterior, this refers to the front end or head of the fish. The mouth, eyes, and gills are located on the anterior part of the fish. And posterior, this term describes at the rear end or the tail of the fish. The caudal fin, which is the main tail fin, is located on the posterior end of the fish. The movement of fish mouth can vary depending on the fishes and feed habits. The way the mandible or the lower jaw and the maxilla or the upper jaw moves as fish opens and closes its mouth might change. Fish possesses maxilla, which remain fixed as the mouth opens and closes. The maxilla is immovable and does not have much dependent movement on its own, while the fish mandible can move forward and downward to open its mouth, enabling to swallow water and catch a prey. When performing a dissection on a fish and inserting a probe through the mouth, it is essential to follow proper dissection technique and handle the fish with care to ensure accurate result. Using a probe, gently open the mouth of the fish to expose the oral cavity. Carefully insert the probe into the mouth of the fish, being cautious not to damage delicate structures such as the gills or the mouth tissues. Guide the probe through the oral cavity towards the back of the throat, applying gentle pressure as needed. Then, apply gently and steady pressure to penetrate the operculum, taking care not to force the probe or cause damage to the gills or surrounding tissues. As the probe passes through the operculum, observe any internal structure or anatomical feature of the gills that become visible. Male's external genitalia consists of a long, slender structure called the genital papilla. So it is located on the ventral side of the fish, just anterior to the anal fin, and it appears as a pointed or elongated structure, often protruding slightly from the body, while the female's external genitalia is typically shorter and more rounded compared to the male genitalia. The African Hito has a sleek, elongated body covered in smooth, scaleless skin. Its coloration can vary, ranging from dark gray to brownish black, often with lighter underbellies. This camouflage helps the fish blend to its environment, providing protection against predator and enhancing its hunting capabilities. The eyes of the African Hito are essential for visual perception. They enable the fish to detect movement, locate prey, and navigate its environment. Vision is particularly crucial in well-lit or clear waters where the African Hito relies on sight to identify food sources and avoid predators. The whisker-like barbels protruding from fish mouth, these are sensory organs, are equipped with taste buds and nerve endings, aiding the African Hito in locating food, especially in murky or dark waters. African Hito has three pairs of barbels: the maxillary barbels, the outer mandible barbel, and the inner mandible barbels. The mouth is equipped with specialized structures, including teeth and the sensory barbels. These adaptations aid in the feeding behavior. The teeth help the fish to grasp and manipulate prey items, and the mouth also serves as the entry point for food, allowing the African hito to consume a variety of prey, including insects, small fish, and plant matter. The operculum is a bony flap covering the gills of the African hito. It plays a crucial role in respiration by protecting the delicate gill filament and regulating the water flow over the gills. It also helps maintaining the proper oxygen levels within the fish bodies, ensuring efficient gas exchange for survivals. Pectoral fins are located on the sides of the fish, behind the gills and just below operculum. They play a crucial role in maneuvering, balancing, and controlling the fish directions and movement. Pelvic fins. These fins are located on the ventral or bottom side of the fish, just below the pectoral fins and typically closer to the tail. Primary involves in stability, steering, and sometimes in reproductive behaviors. Dorsal fin, located on the fish backs, provides stability and balance to the fish while swimming, helping to prevent rolling or tipping from side to side. Anal fin is located on the ventral or lower side of the fish, near the posterior end of the body and just anterior to the tail fin. Like the dorsal fin, the anal fin contributes to the stability and balance during swimming, helping to maintain the fish trajectory and orientation. At the rear end of the fish, we find the caudal fin, also known as the tail fin. This powerful fin propels the African hito forward through the water with precision and speed, essential for evading predators and catching prey. When dissecting an African hito and removing the gills, it is important to do so carefully to preserve the integrity of the specimen and ensure a thorough examination of the gill structure. First, identify the operculum, which is the bony covering protecting the gills. It is located on the either side of the head behind the fish eye. So with the use of scissors, carefully make a cut along the edge of the operculum and gently lift the cut edge of the operculum using a forceps or a probe to expose the gills beneath. So once the operculum is open, observe the gill arches and filament. So using a dissecting scissor, forceps or hand to carefully detach the gill from the gill arches. Start at the posterior end of the gill arches and work towards the anterior end, taking care to avoid damage to the delicate structure. Cut any connective tissue or ligaments holding the gills in place, ensuring that they are fully detached from the gill arches. So when it is removed, examine the gills closely for any sign of damage, parasite, or abnormalities. Also note of the size, shape, color of the gills as well as observable features such as gill shriekers or blood vessels. Parts and function of gills. The African hito, like other fishes, relies on its gills for respiration, which involves the exchange gases like oxygen and carbon dioxide. The gill arch is a skeletal framework that supports the gills. It provides attachment point for the gill's filament and gill's component. Our bony structure or cartilaginous projection found along the inner surface of the gill arches. Their primary function is to filter out particles suspended in water such as debris, planktons, and other organic matter. Gill filament are thin finger-like structure arranged in rows on the gill's arches. They provide a large surface area for exchange of gases between the fish blood and its surrounding water. Each gill filament is covered with numerous gill lamellae, which are thin, leaf-like structure. These lamellae further increase the surface area available for gas exchange. Their thin membranes allow the efficient diffusion of gases across the respiratory surface, ensuring that oxygen is absorbed and carbon dioxide is eliminated effectively. Dendritic organ is specialized structure found in the gills of some fish species, including African Hito. It is rich in blood vessels, which allow for increased oxygen uptake from the water and facilitate the transport of oxygenated blood throughout the fish body. These help ensure that the oxygen is efficiently distributed to the fish tissues to support its metabolic processes and overall health. The presence of the dendritic organ in the gills of the African Hito may confer a greater resilience to the environmental stressors such as pollution or fluctuation in water quality. When performing a dissection of African Hito, ensure you have all the necessary tools and equipment, including tray, scissor, forceps, and pins. First, rinse the external surface of the fish with water to remove any debris or contaminant, and place the African Hito dorsal side up on the dissecting tray. And use dissecting scissors to make a longitudinal incision along the midline of the ventral surface of the fish, starting from the anus and extending towards the operculum. Take care to avoid puncturing internal organs while making the incision. Next, gently open the body cavity by spreading apart the incision, and also take note of the position and appearance of the internal organs. The internal anatomy of African Hito. Like all fish, it has a well-developed internal organ system designed for swimming, feeding, and reproduction. The heart of the African Hito, like other vertebrates, it consists several parts that work together to pump blood throughout the fish body. So we have the atrium, the upper part chamber of the heart, which are responsible for receiving blood from the fish body. Also the ventricle, the lower chamber of the heart, responsible for pumping blood out of the heart into the fish gills where it receives oxygen. Liver performs various metabolic functions, including the synthesis of protein, storage of glycogen, a form of energy, and a metabolism of fat and carbohydrates. It also plays a crucial role in detoxifying harmful substances such as metabolic waste products and environmental toxin from the blood. Gallbladder plays a crucial role in digestive process by storing and concentrating bile, a digestive fluid produced by the liver. So bile contains bile salt, which aid in the emulsification and digestion of fat in the intestine. The stomach of the African Hito plays a crucial role in initial stages of digestion. It receives food from the esophagus and mixes it with the gastric juices, including the hydrochloric acid and digestive enzyme. While the intestine of African Hito are responsible for digesting food and absorbing nutrients. Through the action of digestive enzyme and acid, the ingested food is broken down into smaller molecules that can be absorbed into the bloodstream and utilized by the body for energy, growth, and repair. Mesenteric fat, also known as the visceral fat, are adipose tissue deposits and located around the internal organs like stomach and intestine within the abdominal cavities. These fat serve as a storage site for excess energy. So mesenteric fat also play a role in buoyancy regulation. So in some species, including the African Hito, mesenteric fat contribute to the fish overall buoyancy by providing a means of adjusting its density. The ovaries of African Hito are oval or elongated shape and also a paired structure. The color of ovaries can vary, but it is often similar to that surrounding tissue, ranging from pale to reddish brown. The surface of the ovary may appear smooth, with visible cluster of ovarian follicles, giving them slightly bumpy texture. So the primary function of the ovary is the production and maturation of eggs for fertilization. So once it is fertilized, the egg develop into embryo and eventually hatch them into larvae and continuing the life cycle of African Hito. The testes of African Hito, they typically appeared as elongated and paired structure. The color of the testes can vary from pale or whitish in appearance, while the surface of the testes is smooth and slightly lobulated. The primary function of the testes is the production of sperm cell through the process of spermatogenesis. Sperm cells are essential for fertilizing egg release by female African Hito during reproduction.